Good morning, Shem Stars. In Chapter 11, Biotechnology Principles and Processes, in previous presentation we discussed about vectors. In this presentation we will talk about competent host organism. Let's understand what is competent host organism. Competent host organism for transformation with recombinant DNA is required because DNA, being a hydrophilic molecule, cannot pass through cell membranes. In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up DNA. Let's understand meaning of competent host. Ability of a host to perform certain task. Here it is referred to as the ability of bacterial cells to become host for foreign DNA. Competent host cells can be animal or plant cells too. Not necessarily needs to be bacteria. Now let's talk about methods to make a cell competent. These are chemical method, physical methods, and disarmed pathogen vectors. Chemical method. In this method, the cell is treated with a specific concentration of a divalent cation, such as calcium, to increase pore size in cell wall. The cells are then incubated with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius and then putting it back on ice. This is called heat shock treatment. This enables the bacteria to take up the recombinant DNA. The next method is physical method, and in this method a recombinant DNA is directly injected into the nucleus of an animal cell by microinjection method. In plants, cells are bombarded with high velocity microparticles of gold or tungsten, coated with DNA, called as gene gun method. And the last method is disarmed pathogen vectors. When allowed to inject the cell, transfer the recombinant DNA into the host. So children, now we have learnt about the tools for constructing recombinant DNA. In the next presentation, we will discuss the processes facilitating recombinant DNA technology. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.